So now what we're going to do is start to talk about pathogens. And in this case, we're going to talk about pathogenic bacteria, all right? So we're going to put bacterial pathogens. Because there are other types of pathogens. So there are eukaryotic pathogens, different types of parasitic cells, fungal pathogens, and then they're the virus, they're viral pathogens. So they're all have certain similarities, and then there's certain differences and things that are unique. So this is focusing on bacteria. I've seen students many times on an exam get confused when I ask about infection specifically for bacteria, and then they're describing all the processes of the viral life cycle, which is not what I'm asking. So keep in mind, we're focusing specifically on bacterial cells that are pathogens. So what is a bacterial cell that's a pathogen? It's a bacteria cell that is usually foreign to the host, and causes disease. And I'm going to put underneath host, I'll put host tissue. So for example, there's some bacteria that typically don't colonize your body. If they're introduced to your body and they have no effect on you, then nothing happens. They're soil bacteria that are completely harmless. They're not part of you. They don't typically colonize your skin or any type of your digestive tract. But if you were to ingest them or get them on your skin, they have absolutely no effect. Those are foreign bacteria, but they're not pathogens at all. There are other bacteria that, when introduced onto your body, then can cause disease. So these are the pathogens. Now, there are also some bacteria that are in your body that are part of your human microbiome, and they belong in certain tissues. So in your intestines, you have E. coli, for example, and it belongs there, and it's healthy to have E. coli there. Although there are strains of E. coli, different versions, that are pathogenic, that actually cause disease. So if you were to then ingest one of those types of E. coli, you then become very ill. So even though E. coli itself, generically, certain strains are part of your biome and are healthy to have, there are certain E. colis that then cause disease, and those are the pathogens. Okay, so that's what we're specifically talking about. Those organisms that cause disease, either they're never there or they are there, but they're versions of those organisms, or they get into tissues that they're normally not found. They're not in your respiratory tract, for example. They're not supposed to be there. So potentially, either they get cleared out of the respiratory tract or they cause problems, and then they're disease causing. So that's what we're looking at. Not all pathogens are equal. Organisms that are pathogens have a certain amount of virulence. Virulence is essentially how deadly or damaging they are, and usually this is based on the number of cells of that organism that can cause death. And so usually there's something called an LD50 value. And so I'm just going to attempt to got a little graph here, mini graph. So we have two species of bacteria. For example, 1 and 2, there's a detailed one in our PowerPoint notes. This is, let's say, an LD50. And it's the number of cells, or this would be like the mortality, right? So this is death. All right, and this would here would be the cell number that causes death. All right, so we may have a small number of cells causing death here versus this organism here, whereas that same small number causes quite a bit more death, all right, in those organisms. Here, we're increasing the cell number, and maybe it goes up a little, but maybe in this one it goes up a lot. All right, so by adding a few more cells, a lot more individuals die, right? So in this particular case, this organism, although maybe these are the exact same number of cells given into the host, this one will kill a lot of hosts, whereas the other one has a smaller percentage that would become very, very ill, right? So that's what we're talking about, virulence. So virulence is really how damaging, so the extent of damage to the host. Typically it involves how fast that happens, where the damage occurs, all these sorts of things are part of this virulence. And so there are things called virulence factors that make the organism, more harmful or less harmful. There is also something, as far as terminology goes, I'll just bring it in right now, called attenuation, which is the loss of virulence in a lab setting. So when grown on a plate, like we would in the lab, there are a number of bacteria that would normally be very dangerous because they produce chemicals that make them virulent or cause a lot of damage to the host. They're toxins that they make. But when grown on a plate, because they're not getting the same chemical signals that they would from their host, they actually stop producing those chemicals. And so they become actually, not completely harmless, but much less virulent, all right, under those circumstances. That's called attenuation, essentially, or the loss of virulence, which can happen over time. So, for organisms to infect their host, for bacteria to infect the host, there are really two major things that are kind of going on here. And we're going to break these down into like some subcategories as well. And I'll break this whole thing down to at least two different short lectures so it doesn't get too long. We're going to talk about invasiveness, how they get into the cell, into the host tissues, how they then multiply in the host tissues, and then the kind of bridges the gap where we start to get into the next one, how they start to cause damage of the host tissue. So then we get into the toxogenesis part, or the actual disease and damage part. So where the organism is both damaging the host tissues for nutrients just to feed themselves, and then also are producing chemicals, all right, that are toxic to the host, and then the host is reacting to them in various different ways. We'll also have a little subtopic I'll get into a little bit about evasion. So while some organisms are invading, your immune system should be starting to react to that and come after them to destroy them. Some bacteria then can respond by evading your immune system, and they have certain mechanisms to do that. So that's kind of the introduction. And so we'll get into the first topic here with invasion, how they do it, and break that down. So we're going to look at our first topic here would be colonization. And so we're going to break down the invasiveness into the colonization, and then something specifically we're going to refer to as invasins, which are going to be chemicals that are going to be used to help penetrate into the tissues. The colonization is really sort of the attachment part. And this can either be specific or non-specific. If it's specific, it's really working in sort of a receptor ligand capacity, where I'll draw this big, obviously the size isn't going to be right to the scale. So this is, say, our bacterial cell. And we're saying is on the surface of this bacterial cell are going to be very specific chemical molecules, usually glycoproteins. And on these cells here, like say, so this is going to represent the host tissue. We have proteins that are receptors on the surface of the cells. And they're meant to bind specific molecules. The bacteria then have, why did I put a T there? It's a D. Ligand, not ligant. I don't know what a ligand even is. So a ligand is the specific molecule binding to a receptor. And this is a highly specific binding process. Okay, so not just any molecule will attach to these receptors, only a very specific molecule will attach to the receptor. Certain bacteria then have evolved to produce these particular ligands, right, these specific adhesion molecules that will allow them to then interact with hosts. Now, this is specific on usually the species level. So I'm talking about the host species. So certain bacteria will attach to these receptors in certain species of organisms, but not others. If others don't have that particular receptor, the bacteria cannot attach to the surface of the cells. In addition to that, it can be even cell type specific. So the bacteria may only attach to certain types of cells. So they may not attach to certain cells in your skin, but they might attach to other types of cells. And then those are the ones that they'll begin this sort of process with. These specific molecules, all right, there's a specific terminology I was going to talk about a little bit here. Generically, they're just called, I guess, adhesins. So adhesins are the ligand. So this is the bacterial part of this. Okay, the part that's actually the bacteria have that's going to bind to the surface receptor. Now remember, bacteria also have other characteristics, right? So many bacteria have sort of this sugar coating sort of on the outside that we call a glycocalyx. All right, so it's a polysaccharide, a sugar layer. That glycocalyx can be organized or disorganized, remember, as a sort of a slime layer or a capsule, and then that can then bind to host cells as well. So it's a little more of a non-specific sticking to the host cells through this sugary glycocalyx layer. It's possible that there's some specific binding to it, but usually not. Some capsules then have specific binding, and there's certain things like S layers and all that will have specific binding, which is now a protein layer. So there's other types of layers outside of the bacterial cell that could also have a function in binding them to the host cell. We also had teichoic acids and the lipoteichoic acids in the gram-positive bacteria. They can be involved in the binding, and then the LPS layer of the gram-negative bacteria can be involved in the binding, as well. So there's a number of different things that are involved in the binding. The most specific ones are particular molecules called adhesins that are on the surface of the bacteria that bind them to very specific receptors on the surface of the cells. In addition to that, the host mucus that lines, say, respiratory passageways, then bacteria can get stuck in that. Now that can have two effects. One is it can destroy the bacteria, or it can actually just provide a nutrient, really rich, a broth-like environment for the bacteria to grow if they can evade, which we'll talk about, the host defenses. Right? And so that they're it's almost like the host is sticking the bacteria. It's like a flypaper for the bacteria, and the bacteria are just stuck there, and then they actually start to grow because they can tolerate that environment. They actually like that particular environment, and then it can lead to invasion, or other problems. Okay. So for colonization is usually specific, and bacteria have specific factors that will identify host and host tissues. But there can also be other types of non-specific binding, which usually has to do with the coatings of the cells. Now, invasion. These molecules are usually enzymes that are going to be released from the bacteria. So once this bacteria then gets attached to the surface of the cell, there's not something else I'm going to talk about. I missed over there. Will initiate the release of certain enzymes that will start to attack and damage both potentially the cells themselves, okay, or the connections between the cells. Okay, so patients can damage connection between the host cells or damage the cells themselves. Okay, so actually, let me just do a slight modification. So we're talking about invasion. These enzymes are often called, and there's an I here, but there's no O, invasins. So an invasin is a name for generically some kind of enzyme released from a bacteria that's involved in invasion of the bacteria into the host cell's tissue. That's what's going on. There are a couple different categories. And there's all kind of different specific types of molecules that do this. There are some really good tables and figures in one of the reading assignments that I have linked to this in a study guide. But I'm going to go over just a couple of the general categories. Right? So we have essentially one category that includes things called spreading factors. And another category, which is where we have cells that are involved in usually lysis or hemolysis. So it's actually destroying the cells. So spreading factors, put over here. One of the first ones, really a primary type of spreading factor, is called hyaluronidase. O-N-I-D-A-S-E. Okay, so a polysaccharide called hyaluronic acid is involved in connective tissue adhesion. So essentially, cells have to bind to other cells in order to form a tissue. And there are all sorts of connections and matrices and all that keep the cells together in that type of tissue. So in connective tissues, also which can often undergo stress, they need a variety of different, often types of connections as well. And hyaluronic acid is an important part of a ground substance that is a matrix that connects those connective tissues and keeps them together and intact. This enzyme, hyaluronidase, that's created by bacteria, so the bacteria, the pathogen, makes this hyaluronidase, and it breaks down this substance that is connecting the cells together. So essentially what's going on here is it's like the hyaluronidase has been released, and now these cells are broken apart, which allows then this bacteria to invade deeper into the tissue. So instead of being attached simply to the surface, now the bacterial cell is getting into the tissue itself, between the cells, not just on the cells. And so hyaluronidase is one of the main enzymes that can do that. Another really well-known one is collagenase. Obviously, it's going to break down collagen, which again is going to allow the bacteria to penetrate deeper into the tissue. And there are a number of other types of spreading factors that will break down tissue connections and allow the organisms, the bacteria, to get into the tissues more so. And I guess the idea is to spread from the initial site of attachment throughout a tissue. Now, while that's happening, they can be producing other sorts of enzymes. All right, so these are the enzymes that are going to be involved in lysis, all right, of the cells. So Bacteria are then going to need nutrients in order to multiply. So the next real step in this invasion process is going to be multiplication. All right, that's the cells are going to need to divide, cell division. The cells, in order to divide, are going to need nutrients so that they can grow. Some of those nutrients are then going to come from the host cells themselves. So the next step here is to release chemicals like phospholipases. A phospholipase enzyme will then cause the breakdown of a cell membrane, and then the cell will lyse or burst, and then the nutrients and all the chemical molecules within the cell will be released into this space, and the bacteria can then use that as a nutrient broth in order to grow. So it can multiply and then start to produce more cells, which can continue to do the same sort of thing and spread more and increase the infection. That's what's sort of going on. So you have those, hemolysis is essentially a phospholipase is just lysing a red blood cell versus just a other type of tissue, connective tissue cell. In addition to those, you're going to then have other sorts of enzymes that are going to be digestive. So they're the bacterial enzymes. So these are from the bacteria. And they're going to then be released after the initial lysis enzymes. Right? These are ones that are going to break down from that cell the proteins, so proteases, nucleases, etc. So things will break down the DNA and RNA so the bacteria can use it for raw material. Break down the proteins into amino acids so they can use it as raw material, and so on. A whole variety of different digestive enzymes that the bacteria will then release to start to use those cells for their raw material, right, and increase the available nutrients they have for cell division to increase their spread. So that's kind of the first part. The first part of bacterial pathogenesis is colonization. Bacteria have to attach to a cell. Largely, it is a specific process. Once they get there, they're going to release enzymes, invasins, generically, things that can actually get them into the tissue. All right, so usually breaking down the connections between the cells in the tissues. So hyaluronidase and collagenase are two examples. There are all sorts of other specific ones you can read about as well. Those are major categories. Those are collectively called spreading factors. Once the bacteria starts to invade the tissue like this, they then start breaking down the materials within the tissues, and then we get additional cellular lysis, and then the breakdown of the contents of the cell itself. Now, meanwhile, while this is happening, the host cell is going to be trying to defend itself. So once we're done with bacterial pathogenesis, which isn't yet, still going to continue on to this with a few more topics, then we're going to go into the human immune response, which is, as this is occurring, you're responding in a variety of different ways with specific chemical molecules, with specific cell types. Some of them are kind of generic responses to any bacteria, and then some of them are responses that you have developed to encounter this very specific, say, bacteria, because you've experienced it before, and now your body has built up a little army of cells to identify it and attack it, and they're already just sitting and waiting. So maybe it'll stop some of this before it even happens. So this isn't necessarily definitely going to happen. If exposed to the pathogen, you may fight it off before any of these things can occur. But in general, this is what is going to occur. So that's the end of this first part here with invasion, and now we're going to talk about how they evade your immune system a little bit, and then we'll get into the toxins that come more so endo and exotoxins.